So this video is going to be about the regulation of gene expression, specifically operons. So an operator is going to be a segment of DNA that functions as the on or off switch to help control the expression of these genes downstream of the operator. So the operon itself is going to be this complex of the operator, the promoter, which is made up of this whole segment right here, and then the genes that are under the control of that operator and promoter. So a repressor is going to be this molecule right here. So this is going to be a molecule that is capable of binding to the operator to prevent the association of RNA polymerase with the promoter and therefore prevent these genes from being transcribed. A regulatory gene is going to be the gene that encodes this repressor protein. So lastly, a co-repressor then is going to be a molecule that binds with the repressor, which allows the repressor to become active and then bind to this operator to prevent the transcription of the genes in the operon. So we have two kinds of operons. We have repressible operons and then we have inducible operons. So in repressible operons, that's going to be an operon that's usually turned on, but then it can be inhibited by particular molecules. So that's why we say it's repressible, is that it's on, but the activity of that operon can be repressed. So an example of that is going to be the trip operon. So in the trip operon, we have this repressor right here. So in repressible operons, the repressors are going to be made in an inactive state and then become activated when some molecule is present. So in the case of the trip operon, the repressor is made in an inactive state, and when tryptophan is present, it will bind to the repressor to activate it so that it can bind to the operator and prevent the transcription of these genes, which are necessary to produce the molecule tryptophan. So the reason that we do this is that if there's tryptophan present from the diet, then the cell doesn't need to spend the energy, one, transcribing these genes, and two, using these enzymes to actually produce tryptophan itself when it's already present. So that's why this operon is repressible, so that when tryptophan is present, it can bind to this repressor and make it active, and then inhibit the transcription of these genes that really are not going to serve any real useful purpose in the cell at that time. So moving on to an inducible operon. So inducible operons are going to be operons that are usually off, but operons that can be turned on whenever a specific molecule interacts with that regulatory protein. So an example of an inducible operon is going to be the lac operon. So an inducer is going to be this molecule that binds to the repressor and then changes the shape of the repressor so that it's no longer able to bind to the operator. So we'll walk through this example and look at these different parts. So we have a regulatory gene right here, which is going to produce this regulatory protein, so that will be our repressor. So in inducible operons, the repressor is produced in an active form, so that when this inducer is not present, the repressor is capable of binding to the operon and suppressing the transcription of these genes. However, when our inducer is present, which for the lac operon is this molecule called allolactose, then it will bind to this repressor, and now it's changed the shape of the repressor. So the repressor is no longer capable of binding to the operator and preventing the transcription of these genes. So now these genes will get transcribed and then translated into these proteins, which are going to encode for enzymes to break down allolactose. And then when these allolactose levels fall low enough again, then they're no longer going to bind to the repressor, so it will become active again, and the operon will be repressed until lactose becomes present again. I hope you found this video really helpful. All images, unless otherwise stated, are from Campbell Biology's 11th edition. Remember that if you are an enrolled Baylor student, we do offer free tutoring on the first floor of the Sid Richardson building. You can schedule a free one-on-one 30-minute appointment, or you can drop in during any of our normal business hours. For more details, visit www.baylor.edu/tutoring.